Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to Bioscience with Shatreen Sawati. And today I'm here with ribosomes. Yes, I'm going to discuss about the structure, about the discovery, about different types of the ribosomes. Maybe you are not familiar with the types of the ribosomes like the mitochondrial ribosome, chloroplast ribosomes, prokaryotic, eukaryotic ribosomes, their differences, and many more. So now I'm starting my lecture. What are basically the ribosomes? Ribosomes are small granules present in the cell. These ribosome, the word ribosome actually derived from the two words ribosomes. Means ribonucleic acid, because ribosomes are made up of the ribosomal RNA in the nucleolus of the cell. In the nucleolus of the nucleolus is present in small dense organelle in the suborganelle in the nucleus. So this is made up of the ribonucleic acids, mean the ribosomal RNA. So this is the word ribo is used, and the zoma, this is the Greek word for the body, like chromosomes and many more, like that. So zoma is for body and the ribo is for ribonucleic acid. Now ribosomes are tiny spherical dense particles about 150 to 200 angstrom diameter that are primarily found in most prokaryotic and eukaryotic, means all types of the cell. Is it clear? Okay. Now the discovery. The ribosome was first discovered by the, you can see here, I'm sharing the photograph of the George Emil Platt, who is also considered as the father of cell biology too. And ribosomes were first observed in the mid-1950s by the Romanian-American cell biologist George Emil Platt using an electron microscope as a dense particle or granule. The term ribosome was first proposed by the scientist Richard B. Roberts in the end of 1950s. Is it clear? Okay, now what is the Svedberg unit? Mostly the students are confused about the S which is used with like the 70S and 80S, which I will discuss later on. So what is S? Now I'm going to discuss the unit of measurement used to describe the ribosomal subunits and the ribosomal fragment is the Svedberg unit. A measure, what is basically the Svedberg unit? This is the measure of the rate of sedimentation in centrifugation rather than the size. Must keep it in your mind. This is what the measure of the rate of sedimentation in centrifugation rather than the size. That's why fragment names do not aid. Like prokaryotic ribosome is 70S and eukaryotic is 80S. But the 70S ribosomes are made up of the 50S and 30S. If you add 50 and 30, then it will become the what? 80S. But no, the case is not here like that. This is the rate of sedimentation. So 50S and 30S make the 70S. So this is what for what? This is not considered as size. This is for using for the rate of sedimentation. Okay. Now the unit means sedimentation of the ribosomal RNA and protein in the nucleolus of the cell. In the nucleolus of the nucleus. Is it clear? So the unit is named after the Swedish chemist Theodor Svedberg, winner of the 1926 Nobel Prize in Chemistry for his work on dispersed system, colloids, and his inventions of the ultracentrifuge. That ultracentrifugation is used for the at that time for what? The ribosomes. Is it clear? So you are familiar with the ultracentrifugation and centrifugation mechanism. What is basically, so the Svedberg unit symbolized as SR. SV is a non-SI metric unit for the sedimentation coefficient. The Svedberg unit offer a measure of the particle size indirectly based on its sedimentation rate under acceleration. That is how fast a particle of a given size and the shape settles to the bottom of the solution. If you're familiar with the centrifugation, you can easily understand the concept of the Svedberg unit, why it is used for the ribosome and so on. Is it clear now? Okay. Now about the location. Ribosome where are located? Ribosomes in prokaryote and in eukaryote both types of the cells present. So it must keep it in your mind, in the eukaryotic cell, ribosomes are located in the cytoplasm. This is the cell, they are located in the cytoplasm, in mitochondria, in chloroplast, and on the surface of the endoplasmic reticulum. That's why the endoplasmic reticulum sometimes considered a rough endoplasmic reticulum because of the presence of the ribosome, and smooth endoplasmic reticulum because of the absence of the ribosomes. Is it clear? So in prokaryotic cell, as you know that all the membrane bounded organelle like the chloroplast, mitochondria, endoplasm absent. So in prokaryotic cell, ribosomes located directly in the cytoplasm. Must keep it in your mind. Is it clear? For the translation for protein synthesis. Okay, that's very interesting to know. Now, the location of the ribosome in a cell determine what kind of protein it make. First. The second one, the number of ribosomes in a cell depends upon the activity of the cell. Most is what is the number of the ribosome in a cell? So its number depends upon the activity, or you can say that the metabolic activity or the function of the cell. Is it clear? On average, in a mammalian cell, there can be about 10 million ribosomes. It's mean the high metabolic activity cells have higher number of the ribosomes. Is it clear? Now, chemical composition. A ribosome is made from the complexes of the RNA, ribosomal RNA, and protein in the nucleolus, and is therefore referred as a ribonucleoprotein. Around 37 to 62% of it is composed of RNA, means the ribosomal RNA. Here the RNA is ribosome, and the rest is protein. Is it clear? That's very interesting to know. And the structure of ribosome now. Each ribosome is divided into two subunits, the smaller subunit and the larger subunit. The smaller subunit which bind to a larger subunit and the messenger RNA. This is the smaller subunit. This smaller subunit attached are bind with the messenger RNA during translation or protein synthesis. And this is the larger subunit, which is different in prokaryotes and in eukaryotes. Is it clear? So larger subunit which binds to the transfer RNA. So transfer RNA here bind here, messenger RNA bind here. And you know that transfer RNA brings the amino acid from the cytoplasm after reading the message of the messenger RNA. You know that messenger RNA contains the codons, which is the genetic code, and the transfer RNA contains the anticodon. I have discussed codon, anticodon in my lecture about the codon and so must go through that lecture. You will get a lot. So both subunits, smaller and larger subunit, are attached with the help of the magnesium ion. Is it clear? Okay. Now you can see very interesting, very amazing structure of the ribosome, large subunit and small subunit. And you know that now you are familiar that the ribosomal larger subunit attached with the, or you can say that has a binding site for the transfer RNA, and this one smaller subunit has binding site for the messenger RNA. So this larger particle consists of the three sites: E site, P site, A site. I'm little bit discussing here. A site for amino acids, P for the polypeptide, means the protein chain, and the E for what? E for the exit site. So the transfer RNA bring the amino acid, pick the amino acids from the cytoplasm and then bring to the A site. And at the P site, these amino acids form, you can say, attached with each other with the help of the polypeptide bond. Then from the E site, the transfer RNA exit. Is it clear? Means after formation of the polypeptide bond, polypeptide chain with the help of the polypeptide bond between the amino acids at the P site. Is it clear? Okay. Now, the two subunits fit together and work as one to translate the messenger RNA into a polypeptide chain during protein synthesis. You can see here a very clear figure. This is the transfer RNA. That transfer RNA bring the amino acids to the A site, and at the P site you can see here the polypeptide chain form, which is the protein, and from the E side this will exit, this transfer RNA, and you can see here this is the messenger RNA with codons or genetic code, and genetic code consists of 3 nucleotides, and the transfer RNA also contain the anticodon. Is it clear? So this transfer RNA pick the amino acid and form the protein during translation. Is it clear? Okay. Now, what is the polysome or polyribosome? Mostly students confused with the polysome. Both are same. Polysome or poly. Poly means many. Zomes mean body. Polyribosome, poly many. Ribosomes, many ribosomes. So what will happen? During protein synthesis, what will happen? When multiple ribosomes are attached to the same messenger RNA. You know that the both unit, smallest subunit and largest subunit, here there is a space between the two subunits. You know that there is a space in case of the, you can see here, when the two subunits attach with each other, there is a space between both smaller subunit and the largest subunit for the passage of the messenger RNA. And what will happen? Many ribosomes come and attach with that messenger RNA, because this messenger RNA contains the genetic code or anticodon for the protein synthesis. And when many ribosomes attach to the same messenger RNA strand, this structure is known as the polysome or polyribosome. I hope so. Now the concept about the polyribosome, ribosomes and the subunit, each and everything is clear upon you people. Okay, now the function, very easy. They help in the protein synthesis. So the ribosome is a complex molecule machine found within cell of all living organisms that serve as the site of biological protein synthesis. You can see here, this is protein synthesis means translation. Ribosome link amino acid together in the order specified by messenger RNA. Ribosomes act as a catalyst. Must keep it in your mind. Ribosome act as a catalyst in two extremely important biological processes. First is the peptidal transfer. For the peptidal transfer means this transfer of the polypeptide chain, and the other one is the peptidal hydrolysis, means the breakdown. Both are performed by the ribosome because ribosome act as a catalyst too. Is it clear? I hope so. The lecture will be clear upon you people. If you have any question, write in the comment box and like my videos and share. Thank you. Till then, Allah Hafiz.